Welcome back to another session of online teaching. In this session, we are going to discuss about a very important aspect, namely double fertilization. This is very important as far as the improvement in the number of plants through sexual method, as well as the nature will be blessed with so many plant species. Let's see how this happens, though there will be some hindrance for this too. Let's study what exactly and how exactly this happens. In this session, you will be able to define all the technical words that you come across, understand the structure of embryo sac, that is a 7-celled 8-nucleate structure, and then you also able to appreciate the double fertilization, the steps involved in that, and finally the fruit and seed formation. Let's start with inbreeding. What is inbreeding? The fertilization, if it occurs within a single plant, then it's called inbreeding. Though this helps in majority of the plants, as this process keeps on happening in generation after generation, there will be something called inbreeding depression. The inbreeding depression will be taking place or happening because of the same genome continuously downloading from generation to generation. So majority of the plants, even though they are bisexual, the autogamy, auto self, gamy fusion, self-fertilization, and geitonogamy results in inbreeding. As I said, the major disadvantage of self-fertilization is the progeny gets weaker and weaker and causes decreased fitness in population. And this is called inbreeding depression. To avoid this, plants encourage or create their own devices to discourage the self-fertilization. Let's see what are those. And the other one is outbreeding. The fertilization occurs between two different plants of the same species, then it's called outbreeding. The genogamy, cross-pollination, results in outbreeding. The plants achieved outbreeding by many mechanisms. First, unisexuality. The unisexual flowers are produced in the same plant or different plant. In monoecious plants with unisexual flowers like maize, cucurbita, castor, it avoids autogamy. Whereas in dioecious plants like papaya, mulberry, it results in xenogamy. What are the hindrances? First one is incompatibility or self-sterility. In this, pollen of the flower has no fertilizing effect on stigma of the same flower. It is because of different chemical secretions. Example, passiflora, potato. Second, dichogamy. In this, male and female sex organs mature at different times. So when androecium is ready, gynoecium is not ready. When gynoecium is matured, androecium is not ready. Hence, there will be no fertilization. Example, China rose, lady's finger, jasmine, custard apple. Third, hercogamy. The anther and stigma are placed at different positions so that pollens are unable to reach the stigma of the same flower because of the height determines. Now, how do you do artificial hybridization? Pollinating the pollen grains of one flower to the other flower of different plants of same species is called artificial hybridization. Why are you calling this as artificial? Since it is created by us. It's one of the major approaches for crop improvement program. In this method, desired pollen grains are collected and used for pollination. The stigma is protected from contamination with other pollen grains. It is achieved by either emasculation or bagging. What is emasculation? Masculine. Masculine means male. Emasculation, removing the male part of it. It's the removal of anthers or stamens from flower bed before it matures. And second one is bagging, process of putting a bag on the stigma. It is the covering of emasculated flowers with a butter paper to prevent contamination with other pollens. So pollen grains will be protected, the genome is protected. Now we study an all-important interaction called pollen-pistil interaction. It is the ability of the pistil to recognize the pollen of the same species. It undergoes post-pollination event that leads to fertilization. If wrong pollen grain, means other species, is landed on the stigma, pistil rejects by preventing pollen germination. So the pistil will not entertain the pollen grain to release pollen tube. The acceptance and rejection is mediated by the chemical component of pollen grain interacting with the pistil. Now, if the pollen grain falling on the stigma germinates and the pollen nuclei travel into the pollen tube, reach the ovary, then fertilization takes place. So fertilization in angiosperms is termed as double fertilization. And double fertilization consists of two things. Syngamy, first one, syngamy, and second one is triple fusion. So syngamy plus triple fusion is equal to double fertilization. As the pollen rays mature, usually containing one vegetative cell and generative cell, they develop into male gamete. In some plants, pollen grain sheds as two-cell condition. In this, generative cell divides into two male gametes as pollen tube forms. Look at the diagram. There is two nuclei being formed in the generative cell, and those two nuclei are entering into the pollen tube. So we call them as male nucleus 1 and male nucleus 2 for our understanding. As the pollen grains land on stigma, it starts to germinate. Now look at that pollen grain on the stigma and look at the formation of pollen tube entering into style. The pollen tube comes out of germ pore. It grows through the tissue of stigma style and penetrates the ovule, as you are able to see in the diagram. The entry of pollen tube into the ovule is from different places identified into porogamy. If the entry of pollen tube through micropyle, as you are seeing in the diagram, then we say porogamy. And second, chalagogamy. It is the entry of pollen tube through chalagal end. And mesogamy, it's the entry of pollen tube through integument or funicle. Sometimes the pollen tube enters through integuments, then we say mesogamy. As pollen tube enters the ovule, the rupture, the tip of that gets ruptured and the embryo sac released into two male gametes. Look at that, the two male nucleus 1, male nucleus 2 entering it. And they are entering through the micropylar region. One of the male gametes fuses with egg and forms diploid zygote. We call this as syngamy. So male nucleus 1 plus functional egg cell fuse together to form a diploid zygote. And this is referred as syngamy. We will name it as fertilization 1. The another male gamete that fuses with two polar nuclei, okay, and forms PN, primary endosperm nucleus. We call this as triple fusion because three nuclei are involved. Nucleus of the male gamete plus two polar nuclei, forming 3N pen, PN pen, primary endosperm nucleus. We call this as fertilization 2. Since there is fertilization 1 plus fertilization 2, we call this as double fertilization. So we say S plus TF. S refers to syngamy, TF triple fusion is equal to DF double fertilization. Keep remembering this. As you are able to see here, look at that, the double fertilization taking place. The zygote is 2N whereas pen is 3N, endosperm is 3N. Primary endosperm nucleus pen develops into endosperm. Look at that. And once the fertilization happens successfully, then post-fertilization events take place. Post-fertilization events include endosperm development, embryo development, maturation of the ovule into seed, and maturation of the ovary into fruit. And endosperm, as I said just now, pen develops into endosperm. The endosperm is a nutritive tissue. It is 3N in nature. 3 sets of chromosomes will be there. So there are different types of endosperms. nuclear endosperm, as you see in coconut, cellular endosperm, as you see in datura, helobial endosperm, it divides unequally, and we can see this in monocots. In pea, groundnut, beans, endosperm is completely consumed by developing embryo. In some seeds like groundnut, castor, endosperm persists mature seeds. Now look at the development of embryo now. In dicots, the zygote divides transversely into upper suspensor. Look at this portion, suspensor. And in micropyle region, in the lower embryo cell, chelagel region, the suspensor cell divides and forms 7 to 10 cells called suspensor. Please identify the suspensor in the diagram. The upper cell of suspensor enlarges and attaches to embryo sac. The embryo cell or embryonal cell divides into 8 cells. The lower 4 cells develop into plumule, which is going to become future stem, and 2 cotyledons, the seed covers. The above 4 cells near suspensor develops into hypocotyle and radicle. Radicle is going to become future root. The fully developed embryo of dicotyledons has an embryonal axis that is differentiated into plumule, 2 cotyledons and radicle. Look at that. To begin with, it's a globular embryo. The globular embryo is transforming into heart-shaped embryo, and the heart-shaped embryo is developing into a matured embryo, and the matured embryo has got a radicle, future root, plumule, future stem, and seed coverings called cotyledons. And the same thing we can see how these are formed. It's through mitosis. Look at that globular stage, hypophysis, meristem, shoot meristem, cotyledons, and getting into torpedo. So in monocots, the zygote divides transversely into 2 upper suspensor cells present in micropyle region and lower embryonal cell present in chelagel region. The embryonal cell divides into globular embryo, forms massive cotyledon and plumule. Plumule of course caused by coleoptile. Identify the portion coleoptile. The single cotyledon is called scutellum. At lower end of amino axis, radicle develops. As I said, radicle is going to become root. It's covered by a sheath called coleorhiza. And finally, the end product of sexual reproduction is seed. It is a developed ovule. So we define fruit as developed ovary and seed as developed ovule. And seed consists of seed coats, cotyledons, and embryonal axis. As you are able to see, the 2 portions, plumule, radicle, seed coat, and then epicotyle, hypocotyle. And when the seed develops, look at the formation of the new plant. The root system, primary root, secondary is going down into the soil. The plumule is developing into a stem, and when the seeds are formed, seeds will be having coverings. If there is 1 covering, then we say monocot. If there are 2 coverings, then in the seed, then we say dicot. So the cotyledons are swollen due to storage of reserve food. The seeds are albuminous or non-albuminous. How do we differentiate this? In albuminous seed, endosperm is not completely used up. In like example maize, castor, sunflower. In some seeds like black pepper, the new cell is also present. The residual persistent new cell is called perisperm. Whereas in non-albuminous seeds, endosperm is completely used up by growing embryo. Example pea, groundnut. The integument of ovule develops into seed coat. The seeds dry up by losing water, and wherever favorable conditions, whenever they are available, the seed starts growing and start germinating. Now fruit, as I said, the developed ovary is called fruit, and the wall of the ovary develops into a fruit wall called pericarp. It surrounds the seed. In apple, strawberry, thalamus develops into fruit wall. Hence, these are called false fruits. Normally, you see, developed ovary is called fruit, but in the case of apple and strawberry, it is not the ovary, it is the thalamus that becomes the fruit. Hence, they are called false fruits. The fruits developed from unfertilized ovary are called parthenocarpic fruits. Already we discussed about the phenomena called parthenocarpy. And I said Bijapur is famous for seedless grapes and seedless pomegranates. So parthenocarpy always results in the formation of seedless fruits. And finally, we come to another phenomenon called apomixis. Here, the seed developed without fertilization of ovule. The seed will be formed without fertilization. The fruit developed from unfertilized ovule is called apomixic fruits. The progeny of hybrids segregate their characters and do not maintain hybrid character generation after generation. Hence, the farmer should buy hybrid seeds every year. It makes them expensive. The apomixis is done in hybrid plants to avoid the segregation of characters. The farmers can use hybrid seeds to raise a new crop year after year. Look at that. The result of apomixis, lot of generations, F2, F3, F4 generations. And finally, polyembryony. The presence of more than one embryo in a seed is called polyembryony. All lemon fruits, citrus fruits, we see this. Hence, in this session, we are able to understand what is double fertilization and post-fertilization developments. I'll see you in the next session.